Thank you all for being here. It's a tremendous showing of support, and I hope a tremendous showing of curiosity. So I hope to answer a lot of your questions and provide you with a little bit of a framework to interpret information you see out there. So this part's really important, the disclaimer. I am not a medical doctor, and this is not even close to medical advice, okay? So please, this is not used to guide medical treatment. These views are my own, not the views of my employer or any other entity, and I'm not paid in any way to make any statements here. This is just something I wanted to do to help people understand this stuff. And this is not a political presentation. Unfortunately, a lot of the ways we talk about the pandemic are political, and this is not the venue for that. So a little bit of background, my degrees are in chemical engineering from UMass, then Northwestern, and then I got my doctorate in chemical engineering at University of Michigan. I studied modulating the immune system with nanomaterials. We're going to learn about the immune system, the innate and the adaptive branches, and how it's used to create vaccines. So I am not a virologist, an epidemiologist, a medical doctor. So you'll see down here in the bottom right-hand corner, that's where the citations are going to be, because I'm going to be presenting the work of experts in this field. So we'll go through the background of COVID. We'll talk about immunity, how it generates naturally, and how we can leverage that for vaccines. Then we'll talk about the spread of disease, the epidemiology, diagnosis, and treatment, and then how we can scrutinize information. And so the goal for this is to provide you with a background that will empower you to scrutinize information that you get in regard to COVID. Hopefully this is valuable and transcends the pandemic. I think it's very useful to be able to scrutinize information. If you zoomed in on the coronavirus, this is what it would look like. And so it's given the name coronavirus because essentially these proteins on the outside, these spike proteins, look like a corona under the microscope. And so the virus itself is called SARS-CoV-2. And so that's severe acute respiratory syndrome, coronavirus 2. The disease that's caused by the virus is called COVID. Distinction is not really important here. We know it has an incubation time of about 4 to 14 days. And so it's a bit sinister for, a week you walk around naive to the fact that you're actually probably spreading it to others. And we know the common symptoms: fever, cold, shortness of breath. And then we know they become a lot more severe in certain patients. So when we think about this in the context of other infectious diseases over the years, we can map them on this chart. On the bottom, it's infectiveness. And so those numbers are indicating how many other people a patient who's infected will spread it to. And then on the y-axis, we have deadliness going up. So COVID-19 is localized with Spanish flu. So that's the flu of 1918. So has a similar contagiousness and deadliness. And you see SARS here from 2003, and then MERS up here being more deadly but less contagious. And then you see some other major players in global pandemics: polio, smallpox, bubonic plague. So that's just contextualizing it. Measles, which we have a vaccine for, is extremely contagious and is way out here on the right side. So here's the anatomy of the COVID virus. And so here on the outside, we have these spike proteins. And so these are very specific and are very targeted to the ACE2 receptor, which is found in the lungs, the kidney, the gut, and blood vessels. This makes it highly efficient at binding to the cells in your respiratory system. So essentially, this is just a package of information. So inside, we call this viral RNA. It's kind of like DNA. And this is the information, the hard drive or the blueprint for making more virus. And so what this essentially is just a cell hijacking replication machine. We know that animals can be reservoirs for disease. So we've had a lot of discussion about bats and other vectors in the news. And so bats have a very special adaptive immune system where viruses can persist for a long time. So essentially the virus won't kill the bat, so it can incubate and mutate over and over before it's spread to these other reservoirs for disease. And essentially humans in certain areas live in close proximity to these reservoirs. We've been able to track SARS-1 through the civet. That was in 2003. 2012, we had the MERS virus in the Middle East through the camel. And there's a few other coronaviruses out there that infect humans, but they're very mild. These are often attributed to the common cold. So coronaviruses are not completely new to us, but there's a lot we don't know and we're learning about. The current coronavirus, SARS-2, we believe came through bat and pangolin, and we'll talk about how we have so much confidence in that. So to really get the most out of this, we're gonna learn about DNA and RNA, just the basics, because that's gonna really help us to understand the really cool stuff and how we can track this across the globe. So here it comes. DNA encodes all genes. So every cell in your body has DNA, and it has the complete set of DNA for every gene in your body. So this is like a blueprint for a huge building or a house. And now that house has many components, and this has all the information for everything. From the DNA, we only express certain genes, and so little bits of RNA are produced to express those genes. The cells in the eye express different genes than the cells in the heart, so they'll just express certain genes with little bits of RNA. So these are like work orders. You know, you'll slowly build this establishment, you'll hire your plumbers, put in a work order for the electricians and the materials, and only what's necessary at the time. And so those RNA work orders are then converted to proteins, and the proteins do work and they provide structure. In this case, your work order produces a crane, individuals to do the job, and structural materials to build everything. And so humans, plants, and bacteria have this whole system, DNA all the way to protein. But RNA viruses like influenza and coronavirus only use RNA and protein. And so here's how it actually works. When the virus enters the body through aerosolization in the air or surface to mouth or face contact, the virus enters and it binds to a healthy cell's ACE2 receptor, which are plentiful in the respiratory system. It binds to that receptor, enters the cell, and dumps out its RNA. And remember, RNA creates protein. So first it actually reproduces that RNA with its own protein material, and it makes many copies, and it starts just generating tons of proteins, which turn into fresh virus particles, and they become mass produced. Essentially, the cell becomes a hijacked factory for replication. And so these virus particles can now enter a healthy cell. Now, the genes in the RNA becomes really important because we can become forensic detectives for the virus. And the reason we can follow it and track it is because sometimes when the virus reproduces its own RNA, it makes a mistake and it causes a mutation. And so these mutations are very easy to spot if you have a sensitive enough machine. And so every 2 weeks, approximately, we find another mutation in this virus. And that mutation might not change how the virus behaves, but it definitely looks different. It's like reading Shakespeare and being very good at picking up typos. And so you have these mutations that pop up. And so when we sample the COVID virus from all these different patients, we can actually sequence them and look for mutations. And so here in blue, we see all these patients, despite where they came from or what time the sample is taken, they all have this blue mutation. And patients A through B have a similar variant mutation, but patients D through E don't share that variant. So what you can do is you can sort out where these patients came from. You can make just like a family tree for the genes of this virus. And so you track it back, you can track it all the way to Wuhan. And so that's what we've done. Some really great scientists have been accumulating all these genetic genomes from samples taken all over the world. And so when we put them together, we can actually track the different variants in these different colors. And so again, what I want to emphasize is it's still the same virus, but there's a small subtle change to it that we can track. If you tracked it all the way back to purple, you'd find it in Wuhan. Yeah, you can actually, these samples are taken all across the world and shared together, and so we can analyze this together. And when I say together, I mean this is a public database, public-private. It's in Germany, and they have over 16,000 shared genomes that are publicly available. So when we talk about, there's a lot of conspiracy theories going around that this was engineered in a lab and shared and spread. The US sent it to China. I mean, here's all the information. It's very transparent, and we can track it all the way back to Wuhan. some people still think it's lab derived, but when we look at the RNA code, so here, this is the RNA for the entire virus, and then we look at the spike, just the outside, the corona, we can look at the sequences there. And here we compare humans to a bat sample to a pangolin. And we see that these are actually proteins that are coded by the RNA. And you see there's these highlighted regions where we see there's highly conserved regions, and we can tell that the humans in certain respects are more closely resembling the pangolin for this spike protein. So you can see this is forensic evidence that it was at least derived from a reservoir that was an animal. And so we see that compared to bat coronavirus, we have a 96% similarity, and with pangolin, 91%. And so it's highly, highly likely that it was derived from an animal reservoir and not engineered in a lab. And actually, there are suggestions that if you were to design this in a lab, you would have designed it completely differently. You would not have predicted it works the way it does based on computer simulation or current knowledge we had. So I also have this question, why does coronavirus mutate so fast? And I get the same question, why does it mutate so slowly? So here's the answer to both those questions. So the RNA is the middleman, it creates the proteins. The coronavirus genome is 3,000 letters. And so that's quite big. In fact, that's twice as big as most RNA-based viruses. So it's twice as large, in fact. And here's the key. Coronavirus has this special spell check protein. So this molecule here, it creates copies of the virus RNA. And when it does so, a normal virus makes a ton of mistakes. And so those mistakes often have a detriment to the fitness of that virus. And so here with the coronavirus, we have the spell check protein, which prevents it from making so many mistakes. And so that's allowed it to create a larger genome over time. So with the smaller viruses like influenza, they don't have spell check, they make more mistakes, higher mutation rate, and that means that every year we have to make a new vaccine for it because it's mutating so fast. With coronavirus, they have this proofreading protein, makes fewer mistakes, has a larger genome, and less drift. And I say question mark, I hope it doesn't drift. And I think we have a high confidence that if we make a vaccine, that vaccine will stay effective despite small mutations. So when we get infected, we have an immune system that protects us a lot of the time. And sometimes it goes awry. But we'll start off by explaining what the two components of the immune system do. The first is the innate immunity. They're the first responders, and they're fast acting. This is like an ambulance or a first responder. They get there, they recognize cell distress, they recognize bacteria and viruses, and they secrete inflammatory molecules, and they clear debris. So if you cut your finger, the swelling, the pus, that's these guys right here. The adaptive immunity is long lasting, and it has a memory, but it's a little bit slower. It takes some training. But when it is trained, you can have actually lifelong immunity. And so this is what we use to create vaccines. So back to the innate immunity, the first responders. There's two major types of cells that are responding here. Neutrophils in green, they're the first responders. They secrete lots of inflammatory molecules called cytokines. This causes cell damage. It also is antiviral, and they're responding to the cell damage and the death. Macrophages, they also secrete inflammatory cytokines, but they also clear debris. So the dead neutrophils will be gobbled up, the dead cells will be gobbled up, and the viruses. And so these work to clear the initial inflammation. The neutrophils are green here because when you get sick and your mucus turns green, that's because there's a ton of neutrophils in it. All right. When you feel groggy, sick, stuffy, that is the immune response making a mess. And so excessive inflammation actually impairs breathing and recovery in COVID. And so this excessive inflammation leads to ARDS, acute respiratory distress syndrome, where essentially the immune cells are building up, creating inflammation, and overreacting. So in a healthy lung, you have oxygen, and that's transferred through a very small barrier into red blood cells to oxygenate your blood. In ARDS, that is filled up with mucus and debris, and it becomes so impaired that ventilators are often used to help support breathing. So adaptive immunity is the learned part of the immune system. They adapt and recognize the pathogen and can create lifelong immunity. And here's how it works. So there's a virus in the body, it's hijacked your cell, and so the innate immunity has recognized the cell distress, and they create all these danger signals. There's a lot of inflammation there. And you have this cell come along, it's called an antigen presenting cell, and it will take up these particles, it will eat them up, digest them, and those spike proteins will end up being presented on the surface of that cell. A T cell will come along and form an interaction. Now we have millions of T cells in the body and they recognize millions of different pathogens. So in this case, a flu T cell would not have a recognition event here and it just floats away. A salmonella T cell wouldn't recognize COVID either. But if a COVID T cell comes along, recognizes the spike protein being presented on that cell, it becomes activated and it divides again. And so this is, the doctor feels your lymph nodes, they're looking for swelling, they're looking for inflammation in all these cells because this event happens in your lymph nodes. So that T cell is subsequently going to find a B cell. B cells have antibodies and it's going to find a B cell that recognizes coronavirus. And when it does, it's going to activate that B cell and it will then produce antibodies, lots of them. And it will continue to produce them until these virus particles are cleared and we recover. And so you notice this is a long, complicated process. There's a lot of mess created by the immune system. Can we skip that ordeal and create adaptive immunity? And the answer is yes. And that is the idea behind a vaccine. Many vaccines are being produced right now to treat COVID, and they all work somewhat differently, but here's what they have in common. They all deliver two things: COVID molecules that are harmless, so these are fragments of the virus or inactivated virus, and an adjuvant. And an adjuvant is a signal to the immune system to be activated. And so here we have the virus particle, very virulent. This is an unmodified virus. What we do is we engineer harmless viruses. We pull out the guts and we make it so that cannot cause disease anymore. But now it has all the proteins that are available to teach the immune system what to attack. So here we have a vaccine or a syringe. It contains fragments from the virus, COVID-2, and has the immune activator. That's taken up by your antigen presenting cells. It goes to the lymph nodes. And again, it will not talk to the flu T cells. It will not talk to the salmonella T cells. It waits for the COVID specific T cells, activates them, produces the B cell, humoral immunity, they call it. And now we have protective antibodies. That was not a very inflammatory process. That's why vaccines don't make people sick. But now we have the protection. And so we understand that some people respond worse to COVID than others. And so we know that worse outcomes are due to dysregulated immunity. And so we have this balance between overactive immunity and well-balanced immunity. So here, when we get too strong of a neutrophilic response, too many macrophages, and they're overzealous, essentially it's like you need an ambulance and you call up 911 and 1,000 show up at your doorstep, and now the ambulance can't get out anymore. And so they had the best intentions but gummed everything up. And they secreted all these factors. So we look in the people who don't do very well with COVID, and we see that all these cytokines are produced, these molecules. And these are associated with worse outcomes. However, in people that do well, you get a neutrophilic response, you get the macrophages, but they don't overreact. They respond early, they clean up, and they go on their way. And we get strong T cell responses that are antiviral and not, essentially T cells have different flavors, but if they have a strong antiviral flavor, then you get better outcomes. So these are the molecules, part of their phenotype. And so risk factors, things that cause dysregulation and hyperinflammation, are age. Unfortunately, as we age, our immune system is not as robust as it used to be. Immunocompromise, so you might be on drugs that change your immune system's function, or you might have an underlying disease like diabetes, hypertension, cardiovascular disease. These things all affect how well your immune system is going to respond. So how long is it going to take to make a vaccine? Typically, it would take 16 years to make one. So that's academic research. That takes a few years. You're doing mouse models and in vitro studies. The preclinical studies, those are going to be in higher order beings, including non-human primates. And then we'll do phase 1 trial, which is primarily for safety. So the FDA and scientists, we care about safety and efficacy. You cannot produce a drug without demonstrating both. And so we take these things very seriously. We don't usually speed up this process. Then you have to build a factory and manufacture the drug. And this is a very arduous timeline, right? We can't wait 16 years for a vaccine. And so we're trying to speed everything up, work very collaboratively between the government agencies and the private enterprise to make this happen very quickly. And Americans are quite industrious people, and I think we're well on our way to doing that. So this is the new timeline. So we've crammed everything in to essentially 12 months to 18 months. And so we're thinking by August 2021, we might be able to have a vaccine. However, there might also be a demand issue where there's not enough vaccines to go around. And so you would roll out essentially who needs it most. It's worth noting that this is going to take a lot of ingenuity. You know, people think about the government and the FDA as red tape, and it is somewhat frustrating when you're trying to push a drug through. But safety and efficacy are paramount to public health. And there have been vaccines that haven't worked that well, and we don't want that to happen, okay? There's some silver linings here. So after MERS and SARS, the government started investing more in researching these coronaviruses. And so they already had some initial work in vaccine development that's kind of giving them a head start. So if someone asked me, does the government really need to play a role in this process? I'll say academic research and government-funded research is kind of nice for these areas that aren't so lucrative. Vaccines are not a great business model. They're not a great money maker. And pharmaceutical companies and innovative biotech companies are both motivated by the payoff, right? They might have a fiduciary responsibility to their shareholders, and a lot of them are venture funded. Vaccines and antibiotics are not very sexy investments. And so that's why it is important to have government motivation behind some of this work. So diagnosing infection we know is very important right now. So it's actually key. When we don't have a vaccine, we at least need to know where the virus is. And so there's two different diagnoses being talked about in the news right now. One is for diagnosing active infection, and the other one's for essentially diagnosing if that person has been exposed and developed immunity, so after disease. So the active infection works by polymerase chain reaction, and this is going to measure if there's actually virus particles in the person. You swab, you convert the RNA from the virus into cDNA, and then you amplify specific DNA from the virus. And so here we have an example. We have a positive person, we know they're infected. We have a negative person, we know they don't have the infection. And then we go through these cycles of amplification. So the DNA exponentially divides if there's viral DNA there, or it does not. So we can see clear as day a positive versus a negative value. And I say clear as day, but not all these tests are 100% sensitive or 100% specific, and we'll talk about that. Now, if we want to know if that person has had a previous exposure and has developed antibodies, so we look along the bottom here, there were virus particles, there was an adaptive immune response, there are now antibodies present, we just have to measure for the antibodies. And so we have these little tests, and so you add a blood sample to the well here, it's kind of like a pregnancy test. They put little spike proteins from the virus onto this assay. Those antibodies will bind to those peptide or protein regions, and that'll tell you if that person has immunity against that specific protein. But there are statistical challenges to these diagnostic assays. So here we are talking about the false negative result. So in this population, we have 100 individuals, 6 of them are currently infected. And we'll use a PCR test to see if they're currently infected. This is a number from the early Abbott test, which is very interesting and promising. 15 minutes, you get a result. It's 85% true positive, meaning that it's gonna capture 85% of your sick patients. And we'll assume it's 100% specific, so no false positives. So out of this group of 100 people, it will correctly identify the 5 people that have an infection, but one person will have a negative result. And so this is a risk because they walk out of that clinic thinking that they're safe, and they go and interact with people and spread the virus more. So even a sensitivity of 85% is not very good. We'd like that around 95%. And then there's the false positive shortcoming. So in the case of the antibody test, who has immunity, we're going to measure antibodies from this patient. Let's assume it has 100% sensitivity. It will track every one of these 6 patients, but if it has a 95% specificity, it's going to include some false positives. A possible outcome here is at the end of the testing, you'll have 11 people who are positive. That is to say, they'll think 11 people had the virus, when really only 6 did. Now 5 people have a false sense of security that they're immune to the disease and won't get it again. And so that's a liability. And so we need to get these diagnostic tests. And 2, we need to make them more specific and sensitive. So there's been a lot of numbers being thrown around saying, things like, oh, as much as 50% of people already had it and were asymptomatic. They're essentially taking small numbers and blowing them up when the number of people who actually have the virus is close to the number of people that are going to be falsely diagnosed. You're going to have a lot of error there. It's essentially as good as flipping a coin in some cases. So what are our management options? We don't have a vaccine, so what can we do? You could do nothing. So unmitigated spread, you have exponential growth of the virus. Or what we can do and what we've been doing is isolating and using hygiene practices. So every time you make a decision that limits your interaction with other people, you cut down a node in this network and you cut down the amount of people that get sick. So if we do nothing, we have a rapid need and demand medically. From the news, our hospitals are completely inundated with sick patients. We do not have a healthcare system in this country that can handle that. And so as long as this line goes into the red region, we're essentially straining our medical system. So that means people that don't have coronavirus, that are ill, which is, hospitals are always busy. People didn't automatically stop getting sick since COVID came around. So people with heart disease, cancer, diabetes, dialysis, having babies, these people also need high quality medical care. And as long as we strain the system, we are limiting the access to quality care. And so it is very important that we do the best we can to keep outbreaks small. So we still have a pretty high daily infection rate. We have to bring that down if we want to return to normal. And so people are talking a lot about herd immunity, and they talk about it almost like it's a strategy. And I would say it's more of an outcome than a strategy. And so here's how it works. In this simulation, all these blue individuals are susceptible to disease. None of them have been vaccinated. None of them have immunity. And so the disease is going to spread completely. And so on the other end of the spectrum, in yellow, we have people who are immune. They might be immune through vaccination or through infection. And so I'm going to click go, and you're going to see the disease spread in red. So we see when a large portion, and that portion is not arbitrary, a large portion of the population is immune, the disease does not spread. You have little flare-ups, but it doesn't spread uncontrolled. And so this is a milestone for public health. So immunity by vaccination, it requires a vaccine, it requires vaccine compliance, and it saves lives. We know this. Herd immunity by infectious transmission, you don't have a vaccine, and the cost of life can be high. So some people say, well, just let nature take its course and let everyone get sick and we'll develop immunity, and this is probably all overblown. That could cost us about 1% of our population, which would be millions of Americans. So as we open up the economy, we got to be very strategic about how we do that. And it's all about operations. The 6-foot rule is a nice rule of thumb. How do people traffic? How do we flow? These have to be operationally controlled. So the herd immunity, it is actually calculated by a simple calculation. So R naught is the basic reproductive number. How many individuals an infected individual spreads to in an unvaccinated population. So for COVID, that's about 2 to 6 people. And we follow the curve up here, and it provides us with the threshold for vaccination that we need to aim for. So that's around 60 to 80%. For measles, which is way more contagious, You follow that up, you see that the vaccination for herd immunity is in the high 90s. So for the government, they set a threshold to reach of 95%. What happens if we don't get herd immunity? This is mumps, measles, and rubella vaccination rate. And this is for the United States over the years 2000 to 2017. And down here we have measles cases. So in 2000, the United States declared that we had eliminated measles in the United States. This is a huge milestone for technology, for medicine, public health. We were actually measles-free declared. And you see that up in the vaccination rate, it's over 95%. So we're where we need to be. But something happened in 1998. There was that infamous vaccine autism paper that was published in The Lancet by Andrew Wakefield. It was found that had data that was deliberately falsified. And so that paper was retracted. No one could reproduce it. But the consequences were great. Essentially, people lost their confidence in vaccines. And so you see that in the United States, the vaccination rate dropped. And so in this red bar, these are the years where vaccination dropped beneath 95%. And so what we found was in those subsequent years, we had measles outbreaks where we had declared the United States eliminated measles, and now we have outbreaks. So this was Disneyland, and then we have subsequent 2019 outbreak. And so this caused governors to make laws making vaccination mandatory. And a lot of people don't like that idea, but this is you can see clear from the data that this is actually a public health idea. If there's too many people that refuse this, then the whole herd suffers, essentially. We've been talking a lot about the 6 feet and the masks and am I safe if I run, things like that. There are scientists out there who have attempted to do simulations with fluid dynamics and particle simulations. And so I've seen these on the news and they're recommending 65 feet or something for bike rides. This paper was not peer reviewed. It was preprint and it made some good press. This is not gospel. The 6-foot rule is a good rule of thumb because particles, the large droplets, sink faster than the light ones. But we know that these droplets can actually go further. 6 feet is just a good rule of thumb. So nothing's absolute. And so when you go outside, when you're running, whatever, there's wind and other factors that this does not account for. And so if you're this guy, though, sneezing like that, these are essentially projectile droplets. They're not small droplets, and this is actually quite bad behavior. In this case, breathing, though, sheds viral particles. And so wearing a mask is the best thing you can do if you're going to be around people. So how are we developing therapeutic options to treat the disease? 12 months, 18 months is a long time to wait. And so I've highlighted here in red all the different parts of the infection and immune cascade that we're thinking about intervening, and drugs are in development for intervening at these points. So we've learned that a lot of people who are susceptible to disease and succumb to disease have hyperactive immunity. Immunosuppressive drugs are theorized to be helpful, but they're not actually advised. Essentially, this whole process relies on immunity, and you don't want to suppress it broadly. However, certain cytokines are very inflammatory and lead to very sick patients. And we can actually specifically inhibit certain cytokines like IL-6, and so that's being tested. We know the virus binds to the ACE2 receptor. Can we modulate that interaction? Can we block it? And so there's peptides and other factors being developed to block that interaction to prevent the virus from going into the cell. And then there's the viral release from the particle into the cell. And so people are trying to target that. Hydroxychloroquine is theorized. We don't actually know how it works, but it's theorized that hydroxychloroquine would inhibit this response. Viral replication, once the RNA is in the cell, it has to reproduce itself. There are clever molecules that will prevent viral RNA replication. Remdesivir falls into this category. And then in the top right, we have antibody plasma. Can we take the antibodies from someone who recovered and infuse them? In theory, it sounds very plausible and reasonable, but we haven't seen that this has worked on COVID-2. We haven't seen compelling evidence from SARS virus either. And then there's the adaptive immunity. It'll happen naturally if you're lucky. We can also develop vaccines that will exploit this pathway. And so we are spanning the gamut. And you see currently there's over 900, this is from a few days ago, there's over 900 clinical studies being conducted internationally to find out what works. And so here in this pie chart in green, about 95% of the treatments are just treatments. They're trying to mitigate the symptoms, the severity. They're not cures. They are not cures. They try to mitigate the damage, help you recover faster. And then about 25 vaccines are in development. Over here, we see a breakdown of the class of drugs that are being repurposed for these clinical studies for COVID. And so we see anti-malarial drugs prominently featured. And malaria is actually a parasite. It's not a virus. And so people have a lot of hope for them, but mechanistically different. We have anti-inflammatory strategies, antiviral strategies like Remdesivir. We have traditional medicine being tested. Antibiotics, again, viruses are not bacteria, but they're trying it. And then blocking cytokines, certain inflammatory molecules. Antiviral drugs, Remdesivir is one. How does it actually work? This is really ingenious stuff here. We saw this molecule earlier. This is what coronavirus uses to reproduce its own RNA. It essentially starts with a strand of RNA and builds a complementary strand. So you see it goes in one way, it adds new nucleotides and builds the strand. What we find is, in this case, this thing is called a nucleotide. It's adenosine triphosphate, and Remdesivir is a mimic of adenosine triphosphate. So you see these molecules look almost identical, but see this odd, weird feature? Triple bond nitrogens don't appear often in nature. When you put it into the body, goes into the cell, you find that it actually incorporates itself and stops the chain elongation. So that virus can no longer produce more copies of its RNA. And so it puts literally, well, figuratively, it puts a wrench in the gears. It's quite selective because, you know, our DNA is not polymerized by this, this construct, but oftentimes drugs have off-target consequences because a lot of these mechanisms are conserved in nature. So we find that Remdesivir is quite selective and we're hopeful, but we've seen mixed results, right? This is from a trial that was done in China. And so it was very well designed. So this is the gold standard, the randomized controlled trial. It's randomized, double-blind, placebo-controlled, and multi-centered. That means multiple hospitals participated. It was stopped early. So the limitation, it's a small study, and these patients were severely ill. This is study time on the bottom, and then improvement rate. What percentage of people improved? We find that Remdesivir in the control really didn't have a significant difference. That is to say, it's really not a miracle drug in this context. And I just showed you the mechanism. Its role is to stop the replication of virus RNA. And we find that the viral load wasn't affected at all in these patients. This is not a promising result. However, the government, National Institute of Allergy and Infectious Disease, they just told us, we haven't seen the data yet, that there's a 31% faster recovery time on Remdesivir. You know, instead of 15 days recovery, it was 11 days, and that was significant. But the survival is, you know, 92% versus 88%. It's not a miracle drug. Yeah, unfortunately. So yeah, going back to hydroxychloroquine, this is the Veterans Affairs study. It turns out hydroxychloroquine actually led to worse outcomes in patients. The death rate was almost 30% with hydrochloroquine alone, 22% with azithromycin, and only 11% if you didn't get these drugs. So we find that you actually doubled your risk or tripled your risk by taking it. And so this was a highly significant outcome. The strengths of this study was that it was an integrated national healthcare system, so everyone's performing very similarly. It was adjusted for confounding variables, but it was not a randomized study, and it was retrospective. We looked back at the data instead of proposing it from the start. And so it's not very promising. It actually looks like this drug could actually lead to worse outcomes for patients. So we see that there are actually consequences to promoting a product without adequately testing it. So that's why we really shouldn't promote them until they've actually shown an effect. This is first-time prescriptions for hydroxychloroquine in the United States over the last year, and we find it's constant. So patients with lupus, rheumatoid arthritis, and other inflammatory diseases are being prescribed this, and it's very steady over time. And then the president made an announcement about this, and it just spiked. And that means that patients who rely on this medication had a difficult time obtaining it. So we really got to look at the science, look at the data, instead of hyping up things before we know their effects. And so that brings me to the conclusion. I really want to talk about how we scrutinize information. So we're constantly bombarded through the internet and TV and everything about information. Some of it's information, some misinformation, some disinformation. When we assess the credibility, we really want to consider who is the author and what is motivating them to share information. What funds them? How are they paying the bills? There are special interests that are, motivating the work. Is there a political bias or motive? And when I take advice or when I think about the information coming from a person, I want to assess their qualifications. What makes them an expert in this field? And is it complete? Did they give me all the information or just the information that provides them with a compelling story? And so I got this nice little diagram. I love the diagram, but I actually read the text from the website it came from. It came from Life Marketing, a marketing agency. And they're giving advice for how to make your posts essentially get more viewership. And they said, make it funny, prompt the reactions you want with emojis. So essentially prompting the emotions, share a surprising statistic, right? How often do you hear about the facts? Some people misuse facts or misunderstand the facts or blatantly lie. And then they say add immediacy with a live video, essentially creating some urgency. People make bad decisions under urgent circumstances. And then ask for the reaction you want. So you see, it's actually quite contrived and manipulative, and this is a lot of how the internet and social media work. So I just want you to assess and ask yourself those questions when you're scrutinizing information. Now, in terms of scientific scrutiny, we use, as scientists, the scientific method. So we make observations, we hypothesize, we conduct experiments that can be measured, and then we confirm the outcomes by other people reproducing it. So here we're looking for measurable evidence. The credibility is built upon the reproducibility of the outcomes and scrutiny by our peers. If our outcomes cannot endure the scrutiny of our scientific peers, then it's not worthy of being part of our knowledge bases. So if your study is so important and so real, it is up to you to convince your peers who are just as expert in the field that it is a real finding. In these pandemic times, timing is critical, it's everything, and so many scientists are reporting their data in preprint publications. These are not peer-reviewed, and so these scientists have the best intentions, but we're finding out after it's already made national news that these studies had limitations, that maybe the statistical analysis was weak, and the conclusions were perhaps wrong. And so the pro is that we have rapid access to information, but the con is that the information may not be accurate. That's kind of showing that the scientific method's working. People publish this stuff. There's an outroar of conversation and scrutiny, and whatever is real and we have high confidence in will endure in our knowledge base.